All right, my darlings, so we have been talking about DNA for quite some time. Deoxyribonucleic acid looks like this. It is a structure that looks like a twisted ladder. It's located in the nucleus, right, of our cells, and it contains our genetic code. And every copy of our DNA in every one of our cells is exactly the same. And so what's the big picture here? Why do we keep talking about DNA? Well, here's the huge picture. In order to be alive, right, if you are a living organism, not just humans, but any living organism, in order to be alive, your body must make certain proteins. The instructions on how to make those proteins are held in your DNA, your genetic code. RNA deciphers the code in the DNA and delivers the instructions to the ribosomes in the cell. Remember all those tiny little dots we looked at in cells? And then the ribosomes read the instructions and build the proteins. So DNA holds the code. I like to say DNA is the keeper of the code, the original code, so we don't lose it, right? It's the original copy. Then RNA comes in and reads the code and then delivers that code to the ribosomes. And then the ribosomes make the proteins that you need to stay alive. So that's the big picture. We have to have protein to be alive, and our bodies make those proteins based on the code in our DNA, deoxyribonucleic acid. So today we're talking about DNA replication. We learned in November how one cell divides into two daughter cells. And so before a cell can divide, it has to make a copy of its DNA. It has to have another copy of DNA to deliver to the daughter cell. So this is just a review here of the cell cycle. We have two daughter cells who have just split through cytokinesis. And if we just follow this one daughter cell, the cell will mature and grow. And then in this phase, we call the S phase of the cell cycle, the cell's life cycle. This is where DNA replication happens. And then once we have a copy of the DNA in the cell will prepare for mitosis and then go through the process of mitosis where the chromosomes or the DNA, right, splits, especially in telophase when you have those chromosomes move to either side of the two new cells. And then cytokinesis happens and you end up with two daughter cells. So if we just follow another daughter cell around, the cell will grow and mature. And then during this S phase right here, notice it says S. During the S phase, we will make, or the cell will make a copy of its DNA so that when it goes through mitosis, one copy goes to one daughter cell and one copy goes to the other. So today we are learning about how the DNA gets copied before the cell goes through mitosis. This phase of the cell cycle is called the S phase or synthesis phase. It happens during the full interphase portion of the cell cycle. Okay, so this S phase, the synthesis phase where DNA is copied, is a quarter, right? You can break this up into 4 parts, 1, 2, 3, 4. So the S phase or the synthesis phase where DNA is replicated is a quarter of the cell cycle. And the cell does it so that it can get ready for mitosis, so that one set of chromosomes can go to one cell and the exact same copy can go to the other cell. So what is DNA replication? By definition, it's the process in which DNA duplicates itself. And if you want to put that in easier terms, it's the process in which DNA makes an exact copy of itself. Why does DNA have to replicate or make a copy of itself? We just talked about it. Before a cell can divide, before it can go through mitosis and divide, it has to make an exact copy of its DNA because one copy has to go with this cell and one copy has to go with the other daughter cell. So when does it happen? Like I said, it happens during the S phase of interphase. S stands for synthesis, and synthesis means to make. So in this phase here, the S phase of interphase, that is when DNA replication happens. So what happens during DNA replication? Let's talk about the steps. Normally, DNA is this double helix shape. It's a ladder, looks like a ladder that's been twisted, right? You have your complementary base pairs make up the rungs. For example, if this were adenine, this would be thymine. If this were cytosine, this would be guanine. Remember, A to T, C to G. So this is what a chromosome or DNA looks like. Sorry, not a chromosome. This is what DNA looks like hanging out in the nucleus of the cell. Before it can replicate or make a copy of itself, it has to unwind. So it has to go from being this twisted ladder to two straight lines. So you would have a normal ladder, right? Two rungs of the ladder. So what happens during DNA replication? Number one, the double helix unwinds. DNA strands usually unwind at several points, creating what we call replication bubbles. So you can see the DNA is unwound here, and the DNA is unwound right there as well. It's all wound up here because you can only see one line, but when it's unwinding, you can see one side of the ladder and the other side of the ladder. Okay. Here's the picture from your course. Here's a section of DNA that is unwound and is ready for replication. When the DNA is ready to replicate, it unwinds into a straight double strand. An enzyme moves along the DNA double strand and breaks the bond between its nucleotides, resulting in two separate single strands. The region of DNA where the strands separate is called a replication fork. So an enzyme will come in, enzymes are proteins, an enzyme will come in and sort of unzip, it'll unzip this DNA strand. It'll break the bond right down the middle between the complementary base pairs. So number two, after the double helix unwinds, the nitrogenous base pairs will pull apart from one another. In other words, we like to say that it unzips. It literally looks like a zipper unzipping. So an enzyme will come in, and the enzyme is represented by this sort of oval, clear oval here. The enzyme comes in and it will move through the DNA molecule, splitting it right down the middle. The base pairs will split apart. You can see that happening here even more as the enzyme moves down, and then unzip a little more and unzip a little more. This section right here where the enzyme is splitting the DNA, we call that the replication fork. It's called the replication fork. Step number three, DNA polymerase, another enzyme, will come in and then fill in the missing bases, right? So we have two strands. We just split this DNA molecule. So here's the strand on the left, here's the strand on the right. They were together earlier. So DNA polymerase, another enzyme, will come in and bring in or haul in base pairs, right, to match them up. So let's look at, we'll look here at this one, G. This is guanine, and DNA polymerase will come in and bring in a cytosine to match it. And then we have adenine here. DNA polymerase will kind of taxi in or ferry in a thymine to match it. Here we have cytosine. DNA polymerase will bring in a guanine to match it. And then I have here a quick video of what happens during DNA replication when the DNA polymerase brings in the base pairs to build up the two strands. So I'm going to click play. It goes a little quick, but I'll show you what's happening. So the DNA polymerase brings in a guanine and a thymine, a cytosine, right? So we're building those two DNA strands. Yes, they're going in opposite directions. And all of a sudden, we have two strands that are exactly the same as the original strand of DNA. I'm going to go ahead and play it again because I know it goes a little fast. Just watch. Okay, so I paused it here. Remember, this clear oval kind of represents the enzyme. And when the enzyme moves up and gets to this nitrogenous base thymine, it says, oh, the matching, base pair for thymine is adenine. So it'll pull in this adenine and connect it to the thymine. And then it'll move up and go to the guanine, and it'll say, oh, the base pair for guanine is cytosine. So it'll go grab a cytosine and match up the cytosine. it gets to this A, adenine, and then we know, of course, that adenine and thymine go together, so the enzyme will go grab a thymine and attach it here. What you'll notice is that the base pairs will move in the opposite direction, right? The enzyme will bring in the base pairs in the opposite direction. So here's the replication fork. The original enzyme came in and split the DNA molecule right down the middle. And then the DNA polymerase is going to come in and bring in the base pairs. Now, on this side, moving away from the replication fork, this is the replication fork. So when the base pairs are added away, moving in the direction away from the replication fork, we call that the lagging strand. And then the strand that gets built toward the replication fork is called the leading strand. So this is the lagging strand getting built, and this is the leading strand getting built. All right, step 4. We made the 2 new strands, right? And they are identical copies of this original strand. And you can tell, right, originally this A here was connected to this T over here. And this C here was connected to this, I'm sorry, that's a G. This G here was connected to this C over here. And we have the same base pairing. Originally this A was connected to this T, and a T came in. Originally this G was connected to this C, and a C came in. Originally this T was connected to this A, and an A came in. So if you look across, you have 2 identical strands, A to T, A to T, G to C, G to C, T to A, T to A. You can do that all the way down the strand, and you can see that they are identical. So at the end of DNA replication, 2 identical copies of DNA exist. Both copies consist of 1 original strand plus 1 newly made strand. So both copies, 1 copy, 2 copies, consist of 1 original strand, and 1 newly made strand, and another newly made strand. Therefore, once DNA unwinds to replicate, it only rewinds with the new strands. The 2 original strands are separated forever at that point. When DNA is replicating, it doesn't do it by itself. There are enzymes that come in and help it replicate. There's more than 30 different proteins involved in the DNA replication process. Each one has a specific job to make sure that the process as a whole is accomplished. And 2 very important proteins in the process are helicase, which unzips the original DNA molecule, and DNA polymerase, which is the one that brings in the new nitrogenous bases. So helicase will work here at the replication fork to unzip the DNA molecule, and then DNA polymerase brings in the new strands. In conclusion, DNA replication results in an exact copy of the original DNA strand. Billions of times a day, cells of your body and of the bacteria living within you divide. Before this can happen, the entire DNA code has to be replicated each and every time. Replication occurs when DNA's double helix opens or unzips. Each strand becomes a template for a new strand. New nitrogenous bases are brought in, and 2 new identical strands are produced. And of course, like I said earlier, there are over 30 different proteins that are involved in the process. 2 important proteins are helicase, which unzips the DNA, and DNA polymerase, which brings in the new nitrogenous bases. So this should look familiar to you. We did this lab last week, I believe. And what I did was already put together one side of the double helix. I'm going to put together the other side of the double helix. Remember, cytosine always bonds with guanine, and guanine will always bond with cytosine. Got to add my phosphate groups. Adenine will bond with thymine always. Here's another adenine, so I will stick a thymine on it. Here's guanine, so I'll stick a cytosine on it. Thymine gets an adenine, A to T. Oops, I forgot my phosphate group. Thymine gets an adenine, and cytosine always gets guanine. And I keep forgetting to put on my sugar phosphate backbone. Okay, so remember, A to T always, C to G always. So here is my DNA molecule, or a section of a DNA molecule. It is not twisted. Okay, so we're going to imagine that it was a double helix. It has now untwisted to begin DNA replication. So I'm going to release the enzyme. So DNA helicase, helicase. Some people pronounce it helicase. You can pronounce it helicase, helicase. It's okay either way. I'm going to release it, and it is going to unzip my DNA molecule. There we go. Now we need to replace the bases, right? Because we split them up. So this one used to be attached to this guanine. Now I need to bring in a new guanine and put it here. This guanine used to be attached to the cytosine, so I need to bring in a new cytosine and put it here and do the same thing to the other side. So DNA polymerase is sort of the ferry that brings in the new nitrogenous bases. So I'll release the DNA polymerase, and it will help me. Of course, it's not going to do it for me, right? It's going to help me put it together. So I had cytosine here. It was bonded to guanine, so I need another guanine. I had guanine here. It was bonded to cytosine, so I got to bring in another cytosine. Adenine bonds with thymine. I have another adenine, so it'll bond with thymine. Guanine gets a cytosine. These two thymines get adenine. Remember, A to T, C to G, and then this gets the G here. So now I have one new strand of DNA, and I'll put this together. This guanine used to be attached to a cytosine, so I'll go grab a cytosine. C to G, T to A, always. T to A, C goes with G, A goes with T. This A is going to go with this T, and this G, right? It used to be attached to a C, and so I'll go grab that C. And now we have a DNA, two new DNA molecules. They're complete, and they can, wind back up into their double helix and hang out in the cell.